what I would concentrate on. 5 attributes of adaptive immunity, definitely. Specificity, inducibility, clonality. Where B and T cells mature. B cells in the bone marrow, T cells in the thymus. The 2 types of adaptive immune response. T cells is cell-mediated, B cells is humoral. Okay? So I think that's important to know. I would like for you guys to know the main parts of the antibody. So I think it would be good for you to know the fact that the arm consists of the antigen binding fraction, or the FAB, and that that arm consists of the antigen binding site. That this small piece right here is a light chain, that this big piece right here is a heavy chain, that we have a hinge, and that we have a crystallizable fraction called the stem. Those are good parts to know about the antibody. I would really like for you guys to know the different mechanisms in which antibodies function. So activation of complement, neutralization, opsonization, killing by oxidation, agglutination, and ADCC. If you guys could elaborate on that just a little bit, that would make me very happy. I would like for you guys to know the main points, the highlights about IgM, IgG, IgA, and IgE. I don't care that you know about IgD. Okay? I'm not going to ask you to list the mechanisms. So I don't want you to know that IgM uses a neutralization and agglutination, and that IgG uses. I don't want you, just the highlights. Okay, just the big picture. So again, the big picture. IgM was a big point with IgM. First one that shows up. What else? What else is interesting about IgM?
Not much of it. It has a high weight.
It has a high molecular weight because it's a
Pentamer.
It's a pentamer.
Mm-hmm.
Good. What was that?
Has a short life cycle.
A short half-life. Very good. Good. What about IgG? What's significant about IgG?
Most abundant.
Most abundant. What else?
And uses every tool to destroy the antigen.
Uses every tool to destroy the antigen in its harbor. Correct. What's significant about IgA?
Inhibits the antibody from being hydrolyzed.
That's by the secretory component. Correct. What else?
It's a
Saliva, breast milk.
Very nice. Good. So most active component in passive natural immunity by going from mother to child via breast milk. What's the significance about immunoglobulin E?
It's rare but important for defense against parasites.
Via which lymphocyte? Eosinophil. Very nice. So those are the main points.
Do they have to know that it's low concentration?
I think, you know what, I think it's a little bit important to know there's low concentration, because next Wednesday when we go over mechanisms of allergies, we're going to find out that one of the problems is going to be a malfunction in the eosinophil, and And so if we end up finding a lot of IgE in the blood, for example, in the serum, that is indicative of assuming that the person doesn't have any parasitic infection, that's indicative of an allergic response. So high eosinophils, high IgE usually implies an allergy of some sort. So it wouldn't be a bad idea to know that IgE usually resides at very, very low concentrations. Okay. What about drinking canal water?
Unless you find out they've been drinking canal water.
Okay. The three types of T lymphocytes: the helper T cell, cytotoxic T cell, and the regulatory T cell. Okay. In the helper T cell, the helper type 1 goes after what? What does it activate? The cytotoxic T cell, correct. What does type 2 activate? B cells into plasma cells, correct. So at least that got named correctly. One goes after the first activated item, which is T cells. 2 goes after the second one, which is B cells. It wouldn't upset me if you guys remember that helper T cells are CD4 and cytotoxics are CD8. So you don't have to remember this entire garbage. But if you could remember those two little clusters of differentiation, that'd be great. And the reason is because you're going to hit in the future. Okay. Okay. This picture I like. That could be on there. So clonal deletion of T cells. This picture I like. It could be on there. Clonal deletion of B cells. I feel terrible, but this is a very heavy chapter. If you could elaborate on the different types of cytokines, interleukins, interferons, growth factors, tumor necrosis factors, and chemokines. So a brief.
What do you want us to know about interleukins?
Okay, what does an interleukin do? Activation of white blood cells. What's the other thing that it does? Clonal expansion. Those are the two main things that interleukins do. It either helps, it is the redundant signal that's necessary to activate a white blood cell, or it is a redundant signal that says it's time to enter clonal expansion. Those are the main roles of interleukins. What's significant about interferon? This is review now.
Activate monocytes.
That's gamma. What about alpha and beta? You guys remember what interferon alpha and beta does? It activates the expression of antiviral proteins to arrest the translation of viral proteins. In short, I call it the Paul Revere because it goes ahead and tells other cells, hey, there's a viral infection. Let's protect ourselves against this. Okay? So as long as you can understand that, as long as you can say it in those terms. This is as bad as it's going to get, okay? This chapter, I think I've been warning you about this chapter for a while. This chapter is horrible, okay? There's a lot of information in it. Growth factors necessary for clonal expansion. Tumor necrosis factors are significant because they do what?
Keeps a check on malfunctioning cells and cancer cells.
Keeps a check on malfunctioning cells and cancer cells. Very good. What are chemokines?
Chemicals released by tissue.
And or other white blood cells to do what?
Communication.
To do what?
Communicate.
Communicate between the cells for signal attraction of the other.
For attraction of other leukocytes to the site. Correct. So that's a layman's term of saying that it is a chemotactic cytokine. Okay. Okay, what's the significance between MAC1 and 2? MAC1 indicates what?
Goes after indigenous antigens, kills infected cells.
That's right. So MAC1 presentation by a cell saying I'm infected by an endogenous or intracellular antigen. I need for this cytotoxic T cell to come over and kill me.
That's, I'm sorry, I got mixed up again. That was 1 or 2?
That is 1. MAC2 is for what?
Antigen presentation.
Antigen presentation. What's the main significance between MAC1 and MAC2 processing?
Is it because it's endogenous?
MAC1 is endogenous, so the microbe is going to be found where? Inside. You can either say intracellularly, which means inside the cell. You can say the cytoplasm. Okay? So it has invaded the cell. Okay? So those proteins will be hydrolyzed in the cytoplasm. Then it will enter the endoplasmic reticulum where they will attach to MAC1 proteins. And then those proteins will go to the surface and it'll say, I'm infected, somebody come and kill me. In contrast, MAC2, what's significant about MAC2? It's exogenous, and the microbe never makes it into the cytoplasm. Never. It stays inside a vacuole the entire time. So it enters a phagosome, which becomes a phagolysosome, where the proteins are hydrolyzed. And then they enter the MAC2. The MAC2 is then presented, and that's happening in an antigen-presenting cell. The 3 main antigen-presenting cells, again, are neutrophils, monocytes, and dendritic cells. Okay. Oh, at the risk of killing you guys, this is a good one. Clonal expansion of cytotoxic T cells. Clonal expansion. Clonal expansion of B cells.
And you want us to know the 2 types, the naturally acquired and artificially acquired?
Yes.
Okay.
Unfortunately. There's a lot of, well, there's just a lot of meat on the bone here, and I'm feeling, I'm not lying to you. I feel really guilty for throwing this much at you. But, I mean, it is what it is.
At least we got the full 2 hours to cover it. You know, we didn't have like an hour and 15 minutes to cover all this.
Yes, right?
And the cell.
Yeah, I like it. Yeah. It would be really nice for you guys to know that the cell-mediated response can take place in 2 mechanisms. Apoptosis or perforin and granzyme. That's good to know too.
And that's it?
Yes, ma'am. I'm done.
Slow down on this one.
This little guy right here.
It can take place in perforin and granzyme.
Granzyme, yes. Oh.
Would you want to show this photo, or would you just want us to know that when you're looking at a cell-mediated immune response?
Yeah, I think I would just ask you
Okay.
the 2 mechanisms. I don't think I'd throw that entire photo on there. There's no reason for it. Okay, I'm done torturing everybody. I find no pleasure in doing this, I assure you. Norma's laughing at me. She does not believe me at all.
She's really going, boo-ah!
Norma is not believing me.